Since ancient times, humanity has been tormented by the question of where the soul is stored. With the advent of the Renaissance, the most progressive part of humanity, scientists, so the most progressive part of humanity began to reject the idea of divine creation in favor of materialism. Everything should have been created naturally and without the help of any god. However, almost every person, including scientists, saw signs of inheritance almost daily with their own eyes. Someone has eyes like mom's, someone has a nose like dad's. The color of skin or hair passed on to children, or a distinctive birthmark present in every member of a certain lineage. From generation to generation, it's simply impossible to ignore such facts. And by itself, it just led to the conclusion that all information about parents is stored somewhere and gets mixed in a certain way during conception. But where exactly and how? If this question is not answered, it is impossible to respect any religion. You are on the Epoch of Mistrust channel with Mikhail Kalmykov, and as you probably already understood, this episode will be about DNA. Well, since on this channel we try to be objective with you, that is, to consider the issue from all possible points of view, then we cannot do without a good conspiracy theory, such a piece of wild conspiracy theory. But before we get to it, we need to understand what exactly we know about DNA and how it was discovered. We begin. So, the history of the discovery goes back to the 19th century. The first to put forward any substantiated ideas about the nature of inheritance was, strangely enough, a monk, and then the abbot of the Augustinian monastery in Brno, a certain Gregor Mendel. In those times, those who could not afford a university education could get it in a monastery. And so, in the period from 1854, during the year of 1856, while studying, Mendel conducted experiments on pea hybridization. Having analyzed the obtained data, he came to the idea of some genetic puzzle of matter which determines this or that trait of the next generation. A little later, developing his idea, he understood that the result is obtained as a combination of several puzzles. That's how they named the genes puzzles back then. But his ideas were not in demand at that moment. And only at the beginning of the century there was a repeated discovery of Mendel's laws. That's when they came up with the term gene. According to observations, it turned out that this should be some molecule located in the chromosomes. Researcher Thomas Morgan developed the chromosomal theory of inheritance as a result. But even before that, in 1886, while studying leukocytes, Johann Friedrich Miescher developed a method for isolating cell nuclei. And in 1889, he managed to isolate a substance with his method, which he called nuclein, that is nuclear, which roughly translates as nuclear from the word nucleon core. Upon studying this substance, it turned out that its properties were quite unexpected and did not match the properties of other proteins he was familiar with at the time. 8 years later, in 1897, the research was continued by Albrecht Kossel, who determined that nuclein consists of two components, protein and non-protein. The second non-protein component exhibited the properties of a weak acid, for which it was named nucleic acid. A little later, he also discovered nitrogenous bases, and Phoebus Levine from the Rockefeller Institute discovered the presence of deoxyribose in DNA. If you don't understand such a complex name, in reality, everything is simple. Ribose is, in essence, one of the types of sugar. Deoxy means that one oxygen atom is missing in the molecule's composition. That is, deoxy means oxygen-free. That's how the name deoxyribonucleic acid, DNA, came about. You could say that genetics started its journey from this point. The substance has been discovered and its location in the cell has been established. Now it's time to study it. Now it was necessary to understand how this substance is structured, how information is stored in it, and for this it was necessary to understand the molecular structure. Physicists took over this stage. Throughout the first half of the 20th century, debates and research continued, with some insisting that protein carried heredity, while others argued it was DNA. At different times, many famous people participated in these debates, from Delbruck to Schrödinger, the very same who introduced them to the world, his famous cat. In the end, it was definitively established that DNA is responsible for the transmission of hereditary traits. Although not everything went smoothly here, different versions of the molecule structure were proposed. It quickly became clear that it should be a spiral, but which one? For example, it was assumed that the spiral is triple, with phosphorus residues in the center of the spiral as the basis. And only in 1953, James Watson and Francis Crick put an end to this question, having photographed a DNA molecule in X-ray radiation. Of course, it was not a photograph in the literal sense of the word, since the wavelength of light is larger than an atom, and we physically cannot see it. However, by illuminating the molecule with much shorter wavelength X-ray radiation, they obtained not even a photograph of the molecule, but rather a shadow of it, and not even quite a shadow, but a diffraction picture. There are many reflections of a wave from one object, and reflections from reflections, and reflections from their reflections, and reflections from the overlap of one wave with another. However, based on the analysis of all this cacophony, it was concluded that it is still a spiral, a double spiral. This photograph is now known as Photo 51, although, complaints a bit later. In any case, we still can't see the actual atoms of DNA in any real photograph. Even with electron scanning microscopes, and even atomic force ones, we still can't provide such an image, because the molecule simply breaks down during preparation for scanning. By the way, how exactly a scanning electron microscope works, and why its data should not be trusted too much, I told in the video, She is Not Constant to You. The link will now pop up in the top corner or will be in the description of the video. Approximately in the same years from 1955 to 1953, another scientist, Erwin Chargaff, established a rule according to which the number of thymine in the DNA molecule equaled the number of adenines, and the number of guanines equaled the number of cytosines. For understanding, thymine, guanine, adenine, and cytosine are essentially the same blocks of letters that the genetic code is written with. From Chargaff's rule, it follows that guanine always pairs only with cytosine, and thymine only with adenine, and together they form complementary pairs. Well, this also means that the total number of nitrogenous bases in the molecule is always even. So it turns out that DNA is a real existing substance, not a hologram as some believe. And although we can't clearly see it yet, we definitely know the composition of DNA. We know where it is located. We also know that it's a double helix, and we know that it's directly linked to the transmission of hereditary information. Let's try to connect this all together. Most of us imagine the DNA molecule something like this. Either like this or like this. Yes, indeed, the scheme is like that, but it's just a simplified scheme. Let's try to create a more complete, more realistic picture. For this, we will model, according to chemical formulas, the most realistic appearance of each brick of this molecule. We will denote each separate chemical element of the Mendeleev table in it. They will be highlighted by colors as atom balls and will have different sizes depending on their atomic mass. The smallest ball is hydrogen, it will be blue. The next one is carbon, it's slightly bigger and has a black color like coal. In life, by the way. Even more nitrogen, it's gray. Even more oxygen, will color it red. And we also need phosphorus. It's the biggest and has a dirty yellow color. Now we make building blocks out of these bricks. Again, everything according to the chemical formula. Let's start with so-called nitrogenous bases. In fact, their sequence is the genetic code cipher carrying information about the organism. There are only 4 of them. This is what guanine should look like. This is what cytosine looks like. They can only connect with each other and no one else. This is the basis of what is called a complementary pair. And yes, the name comes from the word complement, meaning these two things are very happy with each other and are always ready to complement each other. But there is also a second complementary pair, and it consists of adenine and thymine. Here it's the same thing. Thymine is ready to enter into a strong relationship only with adenine, and accordingly, adenine only with thymine. As can be seen from the scheme, the connection occurs with the help of hydrogen atoms. It acts as a kind of glue, connecting larger atoms and molecules with each other. That's why such a bond is called hydrogen. Approximately in this way, nitrogenous-based molecules connect with each other. But in order to get a code, a huge number of such complementary bonds need to be written into a chain, a very long chain. And since we are at the molecular level with you, then as threads or bases for recording, logically some molecules should also act. And they must have some properties, but at a minimum, they must be clearly distinguished from the nitrogenous bases themselves, at least so as not to accidentally confuse the lines of code with its carrier when reading. Ribose acts as such a carrier. This is one of the types of sugar. A physicist or chemist will definitely correct that it's a pentose sugar, but for us, a simple definition is enough. Although it's important that this ribose sugar molecule exists in 2 forms, straight and ring-shaped. Joining with a nitrogenous-based molecule, they together form a nucleotide. You've probably heard this term somewhere. Now we have a complete line. No, perhaps the letter of the DNA code, at the center of which are 2 coding elements, connected to each other by a hydrogen bond, and sugar molecules are connected to them at the ends. Now you need to make a lot of such letters and connect them into a chain. And for this, we will need another phosphate molecule, the residue of phosphoric acid, a phosphorus atom to which 4 oxygen atoms have stuck, well, and 1 more hydrogen atom. We connect everything in sequence, and we get a DNA molecule. A very, very long DNA molecule, about 4.5 centimeters long, and all 4 centimeters are packed into a tiny chromosome, which is located inside the cell. By the way, this is one of those moments when popularizers manipulate information for the sake of big numbers. After all, many of us have heard that a DNA molecule reaches 2 meters, 4 centimeters? But the thing is, there's not 1 DNA molecule in a cell, but 46, and each is packed into its own chromosome. Their total length is exactly 2 meters. But this is not the length of 1 DNA, but 46, which indeed are all located in 1 cell, in each cell. However it may be, now the second task is purely practical: decrypt the code so that you can use it for your own purposes later. Well, to correct something, for example, the lifespan, or to cure, or on the contrary, to make yourself slaves. Everyone has their own goals, but in any case, you first need to decipher the code. And how to do this? The problem is actually not a joke. See for yourself. Firstly, it is necessary to extract the DNA itself from the cell. And this is not very difficult. It can even be done at home. The next step is to break it down into its constituent parts and calculate the number of molecules of each type. This is much more difficult, but also possible. For example, to identify those or other foundations, markers exist, including color ones. But what will we end up with? Only a number of letters. Imagine that you took Tolstoy's War and Peace and broke it down into letters. You would get a number of 2,374,000 letters. Could you, knowing the number of letters, find out what the novel is about? Of course not. So at a minimum, words are needed, and such words do indeed exist. They are called codons. It's a part of DNA that contains a sequence of 3 steps, 3 bonds, 3 complementary pairs in a row, if you will. And there are already such combinations. That is, now we have words of the genetic alphabet, codons, but even that is not enough. If we take the same example with the novel War and Peace, we know that there are 467,807 words used. But having all these words, we are unlikely to be able to compose a novel from them. The most we can learn in this way, for example, are the names of some of the main characters. Okay, not some, but all, but only the names. If it comes down to even surnames, confusion will immediately arise. For example, Pierre Bonaparte or Napoleon Kutuzov could very well appear. For a full understanding of the code, it is necessary to arrange all the codons in the correct order. But how to do this? It's impossible to examine individual atoms, not to mention the fact that you need to look through 2 meters of molecular information, molecule by molecule. It was unclear for a long time. And in 1980, Frederick Sanger proposed a rather clever method of controlled division of DNA into small fragments, for which he received the Nobel Prize. In essence, the method involves cleverly marking certain parts of the DNA. The process itself is very complex and lengthy. During this time, the entire DNA molecule is stretched like a cloud overhead, breaking the hydrogen bond. And then, with the help of a special enzyme, so-called DNA polymerase, new complementary DNA strands are synthesized several times. Thanks to the fact that nitrogenous bases take only one previously known element from the complementary pair out of 4, the new chain becomes an exact copy of the template, but as if mirrored. The material for construction is taken from the surrounding solution by polymerase. That's why at this moment, into this very solution, from which the DNA duplicate is formed, specially marked for this purpose, radioactive nucleotides of one type are added. In this way, we literally send our agents into the DNA. The polymerase, creating the complementary part of the chain, adds the radiation-marked molecules into the overall chain. And now there is a possibility to divide one long molecule into relatively short segments of about 500 to 1,000 nucleotides each. Then, with the help of electrophoresis and thanks to the assistance of previously inserted radioactive agents, they are separated. That is, they are placed in a conductive liquid, where under the influence of an electric field, whole pieces of molecules are separated according to their charge. This is a very complex process, but nonetheless quite understandable. And as a result, instead of a chain of billions of molecules, we get quite manageable pieces of a few hundred nucleotides, the sequence of which we can fully read. As you've already understood, this method used to be incredibly complex and lengthy. But the process does not stand still, and at the moment there are many automatic sequencers, but even their execution time for one test can reach up to 2 weeks. And such devices are not cheap, costing on average $500,000 each. Funny detail, despite all the complexity and costliness of the process, If you suddenly need to run a test, for example, on a well-known disease, then a fairly cheap sequencer has been developed for this purpose. It's like $3,730. It costs many times cheaper, only about $18,000. And the analysis time for it is on average about 20 minutes, maximum 3 hours. I am tormented by vague doubts. Well, all right. So we figured out with you how the sequence of the gene is read. Now it wouldn't hurt to understand what is actually written there. But actually there is nothing particularly new here. By the method of simple enumeration, the method of cutting the molecule at a strictly defined place was invented first. In the early stages of this issue, scientists were helped by special restriction enzymes capable of recognizing short nucleotide sequences and cutting them, and other enzymes called ligases, allowed to sew modified DNA back together. This method is still used when it comes to the simplest single-celled organisms. However, if it comes to higher multicellular organisms with much more complex and long genomes, this method worked poorly. Firstly, because the molecule is too long, there are thousands, tens of thousands of parts on it. And if we want to make a cut in a strictly specified place, we first need to select a special enzyme that makes a cut exactly where it is needed. It's not hard to guess that in such a case, tens of thousands of enzymes need to be synthesized. And they are all different. In general, the task is currently impossible. Therefore, the research continued, and by the year 1996, the first method was finally obtained that works stably with the DNA of higher organisms. The so-called zinc finger method. It allowed precise action on strictly defined parts of the genome, working with so-called protein domains of zinc fingers. But the method had a rather unpleasant side effect. In addition to the necessary cut, it gave a certain amount of random cuts in spontaneous places of the chain. Soon, a more promising method of cutting based on chimeric nucleases, called talons from transcription activator like effector nucleases, appeared. Yes, it sounds like a demon summoning spell, so I promise not to say it like that anymore. In 2012 to 2013, finally, a revolutionary breakthrough occurred. A method was developed for manipulating specific parts of DNA using the cell's own mechanisms and its defense mechanism against viruses. Essentially, if you strip away all the unnecessary verbal husk, a necessary virus was created, carrying the required piece of ribonucleic acid, which it then introduced into the cell. From there, the cell did everything itself. In essence, we used an already existing natural mechanism, and with such a syringe in the form of a virus. Yes, in general, a classic virus is a syringe with contained ribonucleic acid that is such a non-living biomachine. Well then, it was time for the most scientific of all methods, the method of scientific poking, or simply the permutation of options. Scientists selectively replaced parts of DNA with blanks and observed the cell. What would happen? What would refuse? Or what new properties of the cell would appear? In fact, this is how they found out where, what, and what is responsible for. So is the DNA code cracked? Yes and no, it's not that simple. Yes, we've broken down the DNA into many small pieces. We know the sequence. In one human cell, there are about 3 billion base pairs, which corresponds to approximately 800 megabytes of information. We know what happens if you replace one piece with another, but that doesn't mean we've cracked the code. In general, the whole situation with DNA is more like hunting wild game. From the very beginning, a predetermined goal was felt. A step to the right, a step to the left is considered a run. Yes, in general, I don't really want to run away, since we've really stumbled upon some kind of Pandora's box, which clearly contains information about a living organism. It's hard to argue with that. The presence of information is confirmed. The direct dependence of the organism on its content is also. The rest is not important. Based on these data, we can already control living cells and even create absolutely new organisms. It works. The remaining questions are already being pushed to the background, and if we look at them, it's purely from a practical point of view. Here's an example. There are 3 billion pairs in a DNA molecule, 3 billion letters that need to be read, and biologists have already found a device for reading information. These are so-called ribosomes. It's also a protein that moves along the molecule and reads what's written there. This same protein also performs tasks. But do you remember our analogy with War and Peace? A huge novel contains 2.5 million letters, and in DNA there are 3 billion. That is, one genome contains 1,300 such novels. It goes without saying that one ribosome will never be able to quickly read all this. That's why there are many ribosomes on the molecule, sometimes even millions. But even in this case, much remains unclear. It is believed that the ribosome reads information from the main DNA, then synthesizes the necessary nucleotide and connects it to the general chain, or creates the necessary protein. However, there are more questions here than answers. First of all, what does reads mean? No, seriously, this is a molecular level. And here we can't just use some general phrases like she looks or she has a frame template. No, it's much tougher here. If information is obtained, it must be in a physical form or in the form of a specific charge, for example. The ribosome itself is about 20 nanometers wide, that is, with dozens of other molecules. Secondly, it must take material somewhere for the construction of the necessary chain. Thirdly, and most importantly, after construction, it must deliver it to the address. And this is the second main question. Genome, but DNA is not the whole cell. It's just a small part of it. And after reading the information, after the necessary piece of protein is built, it is necessary to properly integrate it into the cell. That is, to literally physically detach from the DNA and float with the manufactured protein in the intercellular fluid, exactly to the address and put it in the exact specified part. Yes, of course, for this purpose, there are so-called transport RNAs, but this does not answer the question. Well, let it not be ribosome, let it be ribonucleic acid. But the task remains the same, to deliver the cargo to its destination and assemble it from it. Well, for example, a cell wall or a nucleus or a new organelle. And if the damage to the organism is beyond the cell's limits, or a completely new type of cell needs to be created, simple division won't help. There's still one question that remains unanswered, namely, how was the very first DNA formed? Well, okay, let it not be DNA, but ribonucleic acid, that is, half of the DNA. There is still no answer to this question. Yes, of course, now in laboratories we can create DNA chains. And there is even a technology for their printing, but for it we use the same technologies as nature does. That is enzymes, accelerators, proteins, and so on. But if we are stripped of all this equipment, then even having all 4 nitrogenous bases at hand, we will not be able to form even a simple complementary pair. They simply do not connect with each other without additional enzymes, and an enzyme, in turn, is also a fairly complex protein molecule. That is, for the very first cell, the very first life to appear, it is necessary for 4 nitrogenous bases of a certain type to form in nature on their own. At the very beginning of the video, I showed that these are not at all simple connections. Well, have you ever encountered, say, thymine puddles in nature or clouds of cytosine? It's clear that absolutely any living organism has plenty of this stuff, but the point is that they are only produced as a result of directed biosynthesis. Well, suppose you found all 4 complex components and poured them into one test tube. Do they combine? No, because as we've already clarified, for them to combine with each other, it's not enough for them to just be complementary. Somehow it's necessary to activate the hydrogen bond. That is, to put it in simple terms, hydrogen has only one electron, and thanks to this, hydrogen has a valency equal to 1. That is, it can only bond with one other atom, no more. That's why nitrogen bases can't connect. The place is simply occupied. In order for a second place to form, the electronegativity of the hydrogen atom needs to increase. The simplest way to add one electron is to form a divalent hydrogen ion. There are other options, for example, to change the usual spherical electron orbital, so-called s orbital, to another one, for example, in the form of a dumbbell. It is called p orbital. In this case, one electron will be as if divalent, that is, its electronegativity will increase. It can attract both an atom from one side and an atom from the other. But both variants cause the strongest dissatisfaction among physicists and chemists, because according to modern understanding, this is simply impossible. And if it were, the bonds would be much stronger and would not break with a simple boil, for example. Nevertheless, there is a connection, but it is very, very weak. However it may be, at the very beginning of the formation of nitrogenous bases, they are filled with ordinary water atoms. But at the moment of connection with another element, the bond suddenly appears, exactly at the right moment. That is, some activation of the process occurs. As if someone is smearing the atoms of water with glue. Well, in reality, of course, there is no glue. But possibly the nitrogenous base molecule, remaining overall electroneutral, turns into a kind of magnet, where the plus is shifted to the oxygen atoms and the minus correspondingly to the hydrogen atoms. And in this possibly lies the answer to yet another question, but more on that later. Let's continue for now. So let's assume that you found all 4 foundations, mixed them in a drill, and somehow got them to connect. That's it. Now we've got life. And then again, we don't. Now we need to find another spoon with 5 atomic sugar, ribose, and then another spoon with the remainder of phosphoric acid. Mix everything again and hope that they blind each other in the right order. No, seriously. Even if one pair is blinded, for the DNA code, we still need to blind them into codons, then into groups and haplogroups. At least get 100 steps. But let's say they got it again. And now we have a ready-made tiny DNA. Or even not, let it be just a half, let it be ribonucleic acid, but it's already life. And again, no. Now, we need someone who reads this information and according to the instructions recorded in it, performs the necessary action. Synthesizes proteins and builds the first cell according to them. That is, ribosomes are needed. And not 1 or 2, but in industrial quantities. Transport, ribonucleic acid, and so on and so forth. The whole construction and installation management with various workers. In addition to that, in the newly formed DNA, there must be, among other things, a construction scheme and the ribosomes themselves. Proteins, enzymes, ribose bases, sugars, ribonucleic acid transports, etc. As a result, getting millions of random combinations on a bare, lifeless planet of all these components. And even by connecting them together in the correct order and getting a working correct DNA, in which the correct information is already recorded, we will achieve nothing. Because it will be equivalent to a pile of blueprints on an empty construction site, no one will do the work. No one will even read it. Well, if you think that's all there is to it, then you're wrong. We've only just started to unpack this topic. To be continued.